Everyone with an interest in NASH, or more broadly, fatty liver disease, surfs up. Season 3, Episode 26 of Surfing the NASH Tsunami, our look at what we've learned so far this year about non-invasive testing, and maybe a little histopathology also, starts now.
This week on Surfing the NASH Tsunami.
Why do we like histology? Well, because when you do a biopsy, you always report steatosis, inflammation, ballooning, and fibrosis. And you can't do a biopsy study and only present 2 of those because the other 2 didn't improve or match what you wanted to see. Whereas with biomarkers, we can measure as many as we like and then only report the 1 or 2 which is positive. As a field, we need to come together and select a defined handful of biomarkers that we agree to always include in trials and always report.
Each biomarker has a unique ability to connect to some of the pathophysiological relevant processes we're studying in these patients, and the overlap of how much they improve at the same time in one patient is really interesting. Histology tells us a number of things. This is something attractive and probably tighter linked to histology improvements than a single one.
Quentin, it's always been my belief that every time you improve the quality of the building block, you improve the quality of what you can build.
This stacking allows you to then go into the equity and diversity of the populations. Not all 3 markers might be the same in a Hispanic community, for example. By having better population size in minority populations, we might drive out different data.
Looking at combination therapy, if we can get this NIT menu set, then I think that would bode well for the future of drug development in NASH. And so we have our homework cut out for us because I think we need to mine the data that's been generated from those sponsors that have been willing to do the NITs, and hopefully those same sponsors would be willing to share that data and let us analyze it and come up with the right number and the right thresholds and the right sustainability and the stacking.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join hepatology researchers and key opinion leaders Professor Jörn Schattenberg and Dr. Stephen Harrison, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and this week's guest, hepatology researcher and key opinion leader Professor Quentin Anstee, as they consider improving practices for using non-invasive testing in drug development this week on Surfing the NASH Tsunami.
We're a little late getting started today, and for reasons having nothing to do with you folks, just in terms of recording. And just for kicks, I tested positive for COVID yesterday, so I don't have much voice. So we're going to go very, very short on the introduction. I will note that Tottenham is playing the Champions League next year, that sadly Liverpool did not win their title. Louise and I were texting about this yesterday afternoon. But that Mo Salah from Liverpool and Heung-min Son from Tottenham are going to share the golden boot, so we all have something to be happy about. Stephen is back from vacation, and we have Quentin Anstee with us today. Hey, Quentin, how are you?
Hi, very well, thanks, Roger. Good to be here.
And good to have you. And Stephen, you came back today. Did I get that right?
I did. I came back this morning, and I can't say I'm quite refreshed yet, although I am invigorated to re-engage.
Now, and for those of you who don't see this podcast or any of its short photos on video, you should know that Stephen is in a tie, which I think is the first time I remember this possibly.
Anytime Quentin's coming on, I've got to step it up a notch. I mean, you know.
Quentin is in an open neck shirt.
I feel I've let myself down now.
And Jörn looks positively casual.
Yeah, apologies for that. And I guess I'm the reason you're running late a little bit. And apologies for that to the crew.
Stephen was only a few minutes ahead of you. Don't worry about it. And Stephen was ahead of you because he and I got confused on something. So everybody except Quentin owns a piece of this, and Louise. So with that, let's just go straight to groundbreakers. One good thing that's happened in the last week, personal or professional. Brave one, go first.
Well, I'll go, I guess. I mentioned this to you before we jumped on. So I learned how to scuba dive this past week, so added another something on my bucket list I was hoping to do in my lifetime, and my 21-year-old son and I learned together. So it was a very rewarding experience. For anybody that hasn't done it, I would highly encourage it. For those that have done it, I'm just preaching to the choir, because there's really nothing negative about it. It's a fantastic experience. So my 29 years in the Army only kept me above the water. Finally, after retirement, I'm able to go below. So looking looking forward to more of those opportunities.
Congratulations, Stephen. It's a great thing to have done. It's one of my very favorite things I've ever done in my life, and Louise knows more about it than the rest of us do. So there we go. Next.
I'll jump in next. Despite losing the Premiership title, we did win the FA Cup final. So I'm going to stick with that, and we've got the Champions League final next weekend. Hopefully we'll take all 3 cups, and I'm not scuba diving anytime soon, unfortunately.
Very good. Next.
Yeah, I was going to salute to Stephen's fitness. I guess scuba diving requires some physical training and fitness, so a salute to that, Stephen. I came back home from a short track and field tournament my little ones were involved in, so that was a lot of fun on the personal side. Professionally, there's a so-called disease management program, DMP, being put up in Germany, DMP for obesity, and I'm still hoping that we'll get a mention of liver disease there, but it's a tough fight and not sure how it's going to turn out.
Okay, Quentin?
Yeah, I was just thinking back, but I think one of the most exciting things I've done this week is finally booked a holiday I've been looking forward to for a long time to the Galapagos Islands. So that's something for later in the year.
You can swim with the turtles.
Well, apparently, yes. So, although hopefully I'm going to stay above the water, not like you, Stephen, but time will...
Just bring flippers and a snorkeling mask. Okay, I guess my good news is Tottenham's in the Premier League. Tottenham's back in the Champions League, which was not something anybody would have predicted a month ago. In fact, the night that Louise and I went out in Barcelona with a certain Arsenal fan, he was exceptionally confident about how all this was going to turn out, but a lot of Arsenal fans are having a tough time today, and we're all celebrating. That's a good thing. And at least so far, mercifully, 24 hours into being COVID positive, all I've got is a sore throat, which you can hear right now. So with that, Quentin and I decided about 2 months ago, I guess, that this was the first week he was available to come join us on the podcast, and we would figure out exactly what to do when we got closer to it. We sat down last Thursday and came up with what I think is really an interesting topic, which generally is to take a look at what we're learning about best practices in use of non-invasive tests in clinical trials and other issues. Quentin's given a couple of talks on this recently, and he'll review some of that. And then the purpose of this session is really for all of us to kind of kick around these ideas and make sense out of them, or make different kinds of sense out of them, and to kind of poke and prod and see what we find. So, Quentin, the floor is yours.
Thanks, Roger. So I think it's going to be a great topic to talk about. It's the idea about what have we learned regarding response biomarkers from the drug trials. And really, as you said, I've given a couple of lectures, one at the Liver Forum and one at the ICFL conference in Vienna recently, touching on this subject. And I think there are about 5 key takeaways that we've learned from the trials so far. What I'd like to do is put them out there and hear what you and Stephen and Jörn and Louise think about those, because they're just concepts that are forming rather than totally formed ideas. And it'd be really good to have that sort of dialogue about them and see what we can think. The first one I'm going to go through quite quickly because we've covered it a lot on the Nash Tsunami podcast before. So the first thing I think one can say is that histological trial endpoints, and especially those related to loss of ballooning, are not sufficiently robust, particularly that idea of loss of ballooning, Nash resolution when it's founded on the complete absence of ballooning. I think that's an endpoint that we're all realizing now is building a house on the sand to a certain extent. But there were a few other things that I thought were possibly also worth exploring. So the next one that I'd put out there is the idea that rapidly attaining an NIT response threshold is not necessarily a guarantee of early efficacy. There are a couple of examples of that I'd put out. So if we think about the most obvious one, which is an ALT drop, and you remember from the Flint study, there was the concept of a 17-unit ALT drop as a marker of early response. But when you actually dig deeper into even that original Flint data, what you see is that the positive predictive value of that is, depending on which histological feature you're looking at, somewhere between about 20% and 50%. So you could argue it's very useful because you've gone from not knowing whether the drug works at all to bringing it within the range of a coin toss, and that is not nothing. But it's still wrong to assume that it's a guarantee of efficacy. The same applies to a certain extent to MRI-PDFF. Once again, these are really useful tools. So this isn't about the fact we need to move away from them or anything along those lines. It's just to consider how we interpret them and what we read into them. The next thing I was going to float, and I'll go through them very quickly and then maybe we can tear them apart one at a time, is the idea that simple indirect biomarker panels like FIB4 really need to be interpreted with caution. So again, if we look at what we've learned from a number of trials, and you could say this about the Regenerate study, you could say it about NGM282, aldoferon, and a number of the others, you see very rapid, very early ALT drops, usually within the space of a couple of weeks. And then the ALT levels plateau after that. And then 6 months down the line, we calculate the FIB4 score, and say, oh, look, it's lower, therefore there must have been a fibrosis improvement. And of course, you probably could have got exactly the same FIB4 score at about 3 weeks in. And I don't think we'd claim that all the fibrosis has resolved at that point. So again, it's just about how we interpret the endpoint. A couple of more positive concepts. The next one is the idea that we don't have a single gold standard test. Histology, non-invasives, nothing is perfect. So what we need to do is take a more holistic view of interpreting them. So I think more and more we need to see those waterfall plots, the number of individuals who've regressed, and also the number of individuals who've progressed in each treatment arm. So you can balance that. And we need to start picking, say, 3 biomarkers: a wet biomarker, maybe the ELF test, a physical biomarker such as VCTE or MR elastography, and then a histological biomarker. And we need to start plotting Venn diagrams and look at the commonality with them, because the cases where you're improving all 3 of those, I think we can have a very high degree of confidence that it's moving. Now, of course, that's a very conservative threshold, and we need to think about how we interpret it, but it's also a very robust threshold. And then my 5th and final point is just thinking about histology for a moment. Why do we like histology? Well, because when you do a biopsy, you always report steatosis, inflammation, ballooning, and fibrosis. And you can't do a biopsy study and only present 2 of those because the other 2 didn't improve or match what you wanted to see. Whereas with biomarkers, we can measure as many as we like and then only report the 1 or 2 which is positive. And so I think as a field, we need to come together and select a defined handful of biomarkers that we agree to always include in trials and always report. So those are my 5 thoughts. Each one of them is slightly unformed, and it'd be great to talk about them and find out where the flaws in the arguments are.
Good points. Quentin, let me follow up and make a statement or maybe ask you a question. I think you commented on FIB4 and the ALT decline, making this something that happens early in the trial and it's difficult to assess down the road. So I think one very important statement is that FIB4 is a diagnostic biomarker and not one that should be used repetitively, in particular in longer trial durations, and not as a response biomarker. So is something we would agree on?
So I think that's where I'm coming at it from, Jörn, exactly that.
First of all, I will just respond to your comment, Jörn. If you go back to where FIB4 was originally used, it wasn't even designed for fatty liver. Right? It was designed for HIV co-infection with hepatitis C, because all liver disease that gets better, so the way in our clinical practice, the way we define improvement in liver disease is when we have an elevated ALT and we normalize it. So every time you normalize an abnormal ALT, you should improve FIB4. I mean, I agree with you. It's more of a diagnostic than it is something that we can follow over time. You could arguably say the same thing for VCTE, except I think where they may have a role if the data is mined appropriately is in a responder analysis, such that you a priori say you have to have an X percentage change in FIB4 or an X percentage change or a specific kilopascal change to be in the responder cohort. At the end of the day, that's what patients care about most, right? They don't care that 40% of the people in this population that had a response improved FIB4. They want to know, in my particular case, if I went from a FIB4 of 2.67 and I dropped it to 2.0, did that 0.67 change equate to something that is meaningful to me? And that's what we call a responder analysis. And I think that's different than just saying the p-value changed for FIB4 from pre to post. So maybe it's taking the same thing and just twisting it around a little bit to say, I'm not sure that we can't use it as a prognostic tool or as a measure of therapeutic efficacy, but the way we currently do it, I don't think is necessarily the most appropriate way.
So I think you touched on something really important there. And thank you for saying that, Stephen, because undoubtedly, if a FIB4 drops, that's not a bad thing. That's a good sign because you're taking through a range of biologically relevant features and you're showing an improvement. What I think I'm saying and what we're seeing from some of these studies is that it doesn't necessarily mean that fibrosis is improved. So the concept FIB4 picks F3 and F4 disease. FIB4 drops, therefore you haven't got F3 and F4 disease. When we're seeing the biochemical changes that drive a FIB4 improvement happening within days of starting a new compound, I think it's very unlikely that it's reflecting a fibrotic improvement so quickly.
Yeah, I completely agree. The same thing for FibroScan, right? We take a patient with hepatitis C that has a FibroScan of 15. I go virus negative at 12 weeks. I bring it back into FibroScan 7. Well, I didn't suddenly just take that patient from F3 to F0 disease in 12 weeks. There's clearly an inflammatory component. I really liked your comment, your point too, where you said rapidly obtaining an NIT threshold, interpret with caution. And maybe I paraphrase that a bit, but I agree with that wholeheartedly. And I wonder if we should begin to look at this as, should it be early change, late change, sustained change? And it's interesting, if you look at the injectables, the hormones, the FGF19s and the 21s, universally, with the exception of BMS, pegbelfermin, if we look at kind of the newer ones, so efruxifermin, pegozafermin, for sure those two, where we have rapid and sustained drops in ALT, that is different than, say, a THR beta that is a little bit more slow to see a change in ALT. And in fact, right out of the gate, it may not change at all. And now at 12 weeks, we're beginning to see a decline, but the decline at week 36 or week 52 is going to be lower than it was at week 12. It's more of a, like the tortoise and the hare phenomenon I've talked about before. Some are rapid and sustained, but they don't continue to fall like the FGF21s and FGF19s, or they're slow and steady. But at the end of the day, if you reach a certain threshold, potentially that is meaningful if there's been a period of time for which that's occurred. If we did a biopsy at 4 weeks, which even I might not be willing to do that, but let's say we did a biopsy at 4 weeks because the FIB4 had dropped, or because there was an X percentage change in PDFF, or because there was an X percentage change in FibroScan KPA, I think we would see something totally different than if we did the same biopsy at the end of 1 year with the same drop in percentage-wise of whatever NIT we're measuring. So I totally agree with you. I think the one exception to this rule that probably is worth a discussion is MRI PDFF, because there we have shown that 12-week MRI PDFF change of at least 30% has been linked to histopathologic improvement at week 36. We didn't biopsy, well, we did biopsy an aldafermin, we did biopsy 12 weeks, and we did have MRI PDFF at 12 weeks. Maybe the issue is, when is the right time to measure an NIT? And if you see a change, it's a meaningful change. And perhaps it depends on the NIT question, and perhaps it depends on the mechanism of action driving the NIT change. It gets kind of complicated, I think.
I think it does, and I think it reflects a maturation of our understanding of the clinical trial paradigm, of how the biomarkers are actually working, and combining that with the mechanisms. You said something there that I really liked, Stephen, which was that point about sustained change and that implication, and almost that you can read a biomarker too soon before it's biologically plausible in some situations, which is an interesting thread to pull on.
Or to put it another way, it has a different meaning if it changes in the first quarter of the treatment versus like the last quarter of the treatment.
Jörn, is it about whether it changes in the first quarter of the treatment or whether it stays changed?
That's the sustained part.
Yeah, which I think is the issue. It's maybe less about when and more about for how long. Quentin, when we talked about this last week, you didn't mention the idea of a Venn diagram, and I'm not sure that's exactly the visualization I've got in mind, but if everything flows, then some things happening early will be predictive of other things happening later. Not perfectly predictive, but predictive. So you would think, because I'm taking this past clinical trial and now I'm starting to think about what happens when you have to treat patients where obviously histopathology is not realistic. Do we have the ability, using this kind of thinking, to build models and say, well, if you see this at 12, then you can believe you're on the right track. Next, if you see this at 32, then you know you're still on the right track, and then you should see this in a year or something like that.
So I think this is really interesting. The Venn diagram idea I was putting out there was very much thinking at the, rather than an early readout, thinking at the end of a study. So you've done your, let's say, 24 weeks, 48 week study. You've got your biopsy results. You've got your basket of biomarkers. Currently, what tends to happen, as we all know, is we present, 23% of people had a one-stage fibrosis improvement in the placebo arm. It was 35% in the treatment arm. Oh, and look, ELF test changed in this many on average, and so on. What I'd like to see is to move it to the individual patient level, much more exacting, and then see, well, how many individuals did we, for example, and I'm just picking these at random, have a histological improvement and an ELF test improvement and a FibroScan improvement, all in that same person at that same point. Because those are individuals I would say you could absolutely bank on having an improvement. Now, the numbers are going to get an awful lot smaller. It's a very conservative approach, which has pros and cons to it. But where you do see it, or 2 out of 3, for example, you would have a greater degree of confidence that you're really moving the needle on those patients.
Let's drill down on that a little bit. Could we take it a step further and say that you have to have a certain threshold of change in all 3 of those? And here's another concept that maybe is worth a discussion to see if we can mine data that we've already generated. Instead of saying a threshold, for instance, of a 30% relative drop in PDFF or a 17 unit per liter drop in ALT, what if we said you had to have that threshold, but it had to be sustained for 6 months? Can we mine that data retrospectively to see if we get a more precise estimate of histopathologic improvement relative to just saying, yeah, we had this many patients hit this ALT number, and you've already mentioned the PPV for that was 20 to 50% for ALT. But what if it were a sustained ALT drop of 17 for 6 months versus 4 months or 3 months, or just at the end of treatment, it finally made it?
So that's an interesting thought. I've not thought of that, Stephen. You're right. If it implies a biological effect and it is sustained, I think that's quite interesting. There are almost 2 concepts here, aren't there? There's the idea of sustained, and then at the end of a study, there's the idea of mutual corroboration of different biomarkers. The mutual corroboration one was the one I was thinking of because it's possibly the most easy to visualize, to reconcile. But the idea that you have a run-in to an effect on an individual patient level, I think will be very interesting to look at as well, with as you say, an X% drop in ALT if it is sustained, for example.
And we could mine that from lanifibranor, we could mine that from Sema, we could mine it from Regenerate, even elafibranor. We could mine that data.
And I think that would be very, very informative, actually, because it would give you that sort of trajectory to a hard, well, to a strong endpoint.
Louise, you were wanting to throw water on it.
No, I'm not going to throw water on it. I might put petrol on it, but I'm not going to throw water on it. So we're moving. What you're describing is moving. We can take a lot of what we gain from hepatitis C, rapid viral response, looking at different outcomes. In my dissertation, I did very rapid viral response, where you had a complete drop of viral load to undetectable, and you normalized your ALT by week 2. 100% of people responded to treatment. So we can mine some of that information. If we look at, people have commented on FibroScan there. FibroScan will do the stiffness, but once you start to drop the fat, you start to drop the inflammation, the stiffness levels out. Whereas going back to put a benefit of a biopsy, the one benefit of the biopsies in trial is we know those patients with their FibroScans or their FIB4s have reached that threshold. And I think that's key. We will see a normalization of ALT in a lot of these patients. But what you should see is the plateauing of the FibroScan as that normalizes to be able to get that base. So you can retrospectively mine that data as well as to where did the FibroScan settle and where did that trajectory take you. Because once you get the cap down and the steatosis levels down, it normalizes out. We see it in alcohol detoxification, where you start to bring down the alcohol content, you stabilize the kilopascals, it is the fat that continues to drop. But the inflammation has been lost once we've lost the ALTs and they've normalized. So is that sort of paradigm of looking at these 3 and mining all of it, can you do that? We did it in hepatitis C very successfully, so theory being that potentially yes.
Yeah, hepatitis C is an interesting one. In many ways, it's different to the sort of disease we're dealing with here, just because of the not one single pathogenic feature, but a more complex constellation of features. I think seeing an ALT drop, as Stephen said at the start, and I completely agree with, is biologically very nice to see and very reassuring. But I'm not sure that early ALT drop, other than being nice to see, is necessarily a guarantee of long-term benefit unless it is sustained and unless it reflects further remodeling of the tissue and changes in the pathophysiological processes. So, but the idea of commonality you're talking about there, Louise, ALT coming down, FibroScan coming down in the individual, I think is something that potentially is going to be quite important and quite persuasive as we try to reduce drug failure rates at the end of trials.
And I think that gets to your Venn diagram. If you drop your ALT by 17 units per liter by 4 weeks into treatment, by 12 weeks into treatment in a 48 or 52-week trial, and then at the end of treatment, that's sustained, and you also have had a 2 kPa change in FibroScan at the end of treatment relative to baseline. So you hit both of those factors, and maybe you throw in a third factor, then relating, we can mine that data. We could say, look, if you have one factor, if you have two factors, if you have three factors, how does that correlate to histopathologic response? So now I see another study that we can do. There's one of the sustainability, and there's another one that says if you hit this factor and this factor and this factor versus 1 versus 2 versus all three, how does that correlate to histopathologic response? And not only that, but that would help inform the FDA about moving away from histology. If we could say with a high degree of confidence that you hit, if you hit 3 of these factors and you hit a magnitude of effect change in each one of those factors, that correlates with a 90% probability of hitting histopathology, the registrational endpoint, we're there.
So exactly, Stephen. I think this is the point. And, to go back to what I said about the positive predictive value of, say, the ALT drop or even PDFF on its own. But what we haven't done is stacked them one on top of the other in the individuals and then looked at the likelihood of having those meaningful improvements. And I think that's the sort of thing we should be interrogating these data sets and modeling.
I've been following you guys a little bit from the outside. I guess really speaks to the point that, each biomarker has a unique ability to connect to some of the pathophysiological relevant processes we're studying in these patients, and the overlap of how much they improve at the same time in one patient is really interesting. I mean, we look at histology. Histology tells us a number of things, right? It's the ballooning, more or less well quantitated, the fibrosis, the inflammation, and not all biomarkers address all points in liver biopsy. So I think of liver histology. So I think it is difficult for one particular biomarker to assess all these changes. And the combination, or as you discussed, let's say the largening of the Venn diagrams, more of an overlap, response going the same way. This is something attractive and probably tighter linked to histology improvements than a single biomarker.
Go ahead, Stephen. You look like you.
I see several studies coming out for AASLD. I mean, it's not too late to submit a late breaker, too late to submit a regular abstract probably, but there's a lot of data that Jörn and Quentin, the three of us, could mine and present. And I like the idea of the sustainability versus an early drop. And I like the idea of stacking because at the end of the day, is it 3 that are better than 1? Is 2 just as good as 3? Do you need 4? Because that also gets to your 5th point, Quentin, which is the biomarkers that should be routinely included in every trial. Well, instead of just throwing a dart against a wall and deciding arbitrarily which ones we should include, let's put some science behind it. Let's determine which ones we should include in every trial. Some may move fat, some may work on fibrosis, but at the end of the day, we have a hierarchy of biomarkers that are not used for diagnostics. They're not necessarily used for prognostics. They're used for monitoring therapeutic efficacy. And they may be different, right? All NITs aren't created equal, and they're not all equal for all 3 contexts of use.
Exactly. So it needs to be a basket of them, and we can get this from some of the existing data sets. The problem is that there aren't very many data sets where a range of biomarkers have been systematically assessed, but it's not impossible to look at it. My inclination is that it's going to be a combination of something that measures a physical property, so that's elastography, whether that's MR-based or FibroScan-based. It's something that speaks to collagen turnover. That may be ELF, it may be ProC3, it could be something else. And then potentially something that looks at a biochemical phenomenon. So something as simple as ALT could be one of the features we build into this.
It could be AST. AST could be as good a marker as ELF or ProC3 for fibrosis.
When stacked, you're quite right. So I think these are some interesting concepts that it's worth kicking around, actually.
You see, this podcast is terrific because it allows us to bring brilliant minds like you onto this to generate provocative conversation that may, at the end of the day, lead to something that changes the field.
First of all, yes, I agree. And part of that's because we start with people like you, Stephen, and Jörn to kick that around. So we've got plenty of really fantastic minds to do that. Quentin, I have a question coming out of my more statistical place. When we say stacked, are we talking fundamentally about what you would find in a stepwise regression, where the third measure might not be the third best independent measure, but it might be the measure that does the best job of predicting what's left after you get the first two out of play?
So that's one way of looking at it. That wasn't quite the way I was meaning it. I'm talking about commonality in the same individuals. So the idea that if you reach threshold X in, let's say, ALT, and then also attain threshold Y in PDFF, each of these gives you a greater degree of confidence that you're going to meet your regulatory endpoint. And so the concept is to look at them that way. This is speculative, Roger. And, we as Stephen has said, we need to tease this apart, and we have the tools to do that.
But your point then is that what we're doing when we stack data is we're scoring individuals. We're not doing probabilities. We're either saying on that person, yes or no. And the stacking of the variables is how we get to the yes or the no.
I think there's a difference there as well in the context that this stacking allows you to then go into the equity and diversity of the populations. And not all three markers might be the same in a Hispanic community, for example. So by having better population size in minority populations, we might drive out different data, because I think having used hepatitis C as an analogy, we very much used the predictive value of what we could get to be able to achieve motivation to carry on and that education. So working it backwards, getting that stacking system, getting something that we know can achieve or show sustainability really acts as a motivation. I would argue that all of these markers have to be something that we can use in the real world as well, so that we can take them out of the clinical trial space into the real care where these drugs are going to be delivered in the future. But we might find differences within the populations with that stacking system.
Thoughts about that? I mean, Louise, that kind of feels a little bit... Now you're talking about needing to be able to score different sets of individuals different ways or stack different variables on them. I'm not putting this exactly right, but I think that's kind of what I'm hearing.
Well, I suppose even simpler than that, when you break down the stacking system to see, so say ALT dropped, FibroScan dropped, and ELF dropped, and there was different section cohorts. Women might have dropped better than men. Is that a bigger indication, for example, in females? What was the age? You can tease out the data afterwards, but I suppose we don't have mass trials of different ethnic populations, and I know the FDA are trying to address that with more inclusion. That's where I'm taking it eventually, that you could argue that it might be AST that drops in a particular community rather than ALT being their third or fourth factor. The data's not been drilled down yet, but the discussion is exciting to think that it could be.
Gender aspects are clearly very exciting, Louise. That's, well captured in all these trials. I agree that the diversity in terms of ethnicities is not quite there yet. I mean, there's going to be a lot of questions around, we use different cutoffs for men and women, the percent change, should it be the same? And then does that hold true for all the three biomarkers you named? And we'll see a lot of them improve, maybe two improve, one doesn't, or not sustain. So there's a lot of questions, as Stephen suggested. I think these more granular analyses, I think we're in the position to do them in the larger phase 2 trials.
Yeah, and I think even in the phase 3, just looking at Madrigal's Maestro Nash, I mean, every biomarker that we could think about has been done, to include serum to run ELF, ProC3, CT1 in a subset, MR elastography, PDFF. Of course, FIB4 can be calculated off of that. FibroScan was done. So that's kind of the gold standard phase 3 trial, where you've got histology, you've got multiple NITs that could be appropriately mined to inform the field coming up behind.
So you're absolutely right, Stephen. I mean, I pulled together this table looking at all the different NITs that have been used in all the phase 2, phase 3 studies that have come out so far. And some of those Madrigal studies are some of the best in terms of the breadth of biomarkers they've captured, which really are going to be an immense resource, both in terms of therapeutics, but also in terms of improving the biomarker endpoint space. I mean, my hat's off to you and them for what you've done to put those together.
The downside of that, of course, is the cost, right? And that's a whole nother discussion about the cost of phase 2 and phase 3 trials in NASH drug development. They're inordinately expensive, and I think the cost of a trial today, married with the uncertainty about endpoint, has led to reduced interest in the field of NASH in general. And, venture capital coming into the field, you see stock prices going down. I think we have to your point, we have to find a stable plateau where we don't need to be on fishing expeditions. We need to be looking at these combinations of biomarkers. We don't need to do more. We don't need to do less. We can fix the price at a trial. And I think once we have some positive results, we get a drug or multiple drugs approved, the pendulum will swing back and we'll be on the road again, looking at combination therapy. If we can get this NIT menu set, then I think that would bode well for the future of drug development in NASH. And so we have our homework cut out for us, because I think we need to mine the data that's been generated from those sponsors that have been willing to do the NITs. And hopefully those same sponsors would be willing to share that data and let us analyze it and come up with the right number and the right thresholds and the right sustainability and the stacking that we've talked about today. Those are all new concepts that we haven't even discussed, in plenary session before. We're having our own little mini AASLD and EASL.
It's got to start with some thought and, kicking the tires on it. And I think this is a really helpful discussion in that respect, actually.
Let me maybe ask Jörn, we've talked about a couple of these NITs. Are there any that we've missed that you think are worthy of discussion? Maybe MR elastography, method, MAST, FAST, Agile 3, Agile 4. Should we include those in this conversation as well, do you think?
Good point, Stephen. I think some of them you're mentioning are compound scores already, combining an MRE with blood-based biomarkers, giving a single number. It would be interesting to see if they outperform the staggered approaches Quentin and you just discussed here. So I think those are not necessarily the ones you want to compare them to. Agree that the MRE, Quentin referred to these as a physical, biomarker, are worth to be explored here. And one we haven't done, and that we haven't discussed, and that hasn't been done so frequently, is obviously the liver multiscan. And maybe some of the ones that as a single standalone marker, like the M30, didn't do so well. But if we think to a combination score, the MAC3, where I think M30 is in there, and Quentin, we looked at that in the Litmus Consortium. Actually, that was a pretty good marker. So you can throw in some of those that has been traditionally a little weaker and pair them. And I think then they can still have value.
So you brought up an interesting point, and one that adds a level of complexity to what Quentin and I were discussing, and that is those combination biomarkers that already exist, looking at those in comparison to a staggered approach as well. So in other words, if you took a FAST or an Agile 3 or an Agile 4 or a method or a MAST, and you looked at that in comparison to if your ALT dropped, plus you had a FibroScan drop, plus you had a drop in ELF or something else, that might be a little bit different than just changing FAST, for instance, which does include a change in KPA and AST, but it doesn't get to the third biomarker that you mentioned, Quentin. So
this is something to think about, because the moment we bake them into an algorithm or into a formula like FAST or MAST or whatever, within that formula, you are actually prioritizing some of the signals over others. So, FAST is heavily weighted towards AST, for example, MAST more towards the MRI-PDFF. So the moment you do that, you're already making the algorithm make judgments for you. It will be really interesting to look at this head-to-head. My suspicion is if you set up a robust threshold for each one on its own, it then puts us in the driving seat about how we actually interpret the change rather than it putting it into this formulaic black box. So that's my gut feeling on it, but this needs to be tested and looked at.
Yeah, I agree. This has been a fascinating conversation, Quentin. Thank you for bringing it up. There's a lot of angles to this I haven't ever thought about. Quentin and Jörn, you made some great points, and Louise as well. I think we have some ideas to take back to the drawing board with. Roger, what are your What are your thoughts?
You're kind of way out of my zone here, but one of the questions I do have, if you go back to the history of how some of the composite scores were developed, right? We've talked on the podcast and we've seen in other places how some of these scores were developed knowledgeably and others were, let's just say, less so. For example, some of the histopathological measures we got not because we knew what they represented, but because we could get people to agree on them. So I'm wondering how robust we feel each of these individual measures is for the test that we're going to apply to it. Because if you start stacking things, then you have to have a certain amount of faith that everything you're going to stack is going to, you know exactly what it's going to do. And what it's going to do is what you want. Now, maybe, in fact, everything we've got is that good. Listening to this conversation, you would think a lot of it is. It's just the one curiosity I've got, Stephen, is that if you start stacking things and each thing you stack is in some way variable, it varies from the target you're trying to get to, that's the kind of error that's going to compound really quickly and make problems. So I don't know, just a question.
So I think that's quite an interesting one. It depends how you combine, how you stack, as you say, Roger. The counter argument is each test has an error bar associated with it. But if you believe that there is somewhere underneath it all a ground truth, the actual correct answer, and each of the tests, biomarkers one considers, is an approximation to that ground truth. Theoretically, as long as you have enough patience, and power is another question here, as you stack, you would actually focus in on the ground truth. So rather than amplifying the error, you'd actually have a commonality where you have increasing confidence that you're attaining the ground truth. That would be my interpretation of it.
Okay, so Quentin, does that presuppose that there is one ground truth? I mean, one of the conversations we have around here from time to time, look, I go back to cancer, which is kind of the first thing I ever learned about, and we used to think there was such a thing as cancer, and then we learned over time there wasn't. That different organs had different cancers, and then one organ had multiple cancers. Does that presuppose that there's a common ground truth? Or can we get to that kind of answer even if, in fact, what we're calling NASH is 5 or 6 different diseases is evidenced by the fact that we've got different modes of action that work in different people, and that we think combination therapy will be a solution?
So remember, right now we don't have good biomarkers for NASH. We've got biomarkers for fibrosing steatohepatitis, where due to the collinearity of fibrosis and NASH, you buy one, get one free. So the most tractable area to think about this, first of all, as a proof of principle, would probably be in fibrosis change. So fibrosis, in histological talk, a one-stage improvement in fibrosis. So, but looking at fibrosis would be the area where I think this could be tested out first and looked at in more detail.
And I wouldn't probably call it 5 or 7 different diseases, Roger. I mean, there's clearly a different dynamic in different patients and maybe different drivers. But we all agree this is inflammation leading to the activation of stellate cells, positioning fibrosis, which in the end leads to liver endpoints and mortality. And here I think the fibrosis readout is just the most robust one. And that's what Quentin also mentioned. Looking at that disease stage, we're in more robustness. I sometimes use the frame, the elephant and the mouse. The inflammation is the mouse moving quick in and out and changing rapidly, and the elephant is the slow guy in the room sitting there and building up slowly.
Thanks, Jörn. That was probably imprecise of me to put exactly that way. I take your point exactly. Go ahead, Stephen.
See, Jörn, you can tell you have little kids because you tell these stories in, ways that are interesting and people want to listen.
It's funny, Stephen, I'm back in the movie Philadelphia from the 80s, and Denzel Washington saying, explain it to me like I'm a 6-year-old, right? There you go.
We actually had I got to tell this story. We had a meeting once, and we had a graphic recorder, okay? She draw me a mouse and an elephant on that, and I'm going to show that, and we'll talk one day, and I carry that along with me for some time. I thought it was a brilliant way to put it.
It's great.
Okay. Anything else we want to cover off on this topic, or we started a little bit late, so we could go to a final question and wrap up if we think we've covered everything we're going to want to do today.
I was just going to ask then, if you theorise that you can get a stacking system, you can apply it to the placebo group as well. So if people were you can predict what the placebo outcomes are going to be if they meet all of those factors. Would that be correct in my way? So that you could say by week X, if you've achieved these, that we think the placebo is going to be a 25% response. We can predict it now, and you can pick. Could you do that?
All right. So I would keep the idea of predicting future response out of it. I think I would keep it as cross-sectional, but the point being that one could apply it to each arm of a study. And so if you were expecting looking at the number of individuals who responded on 3 separate biomarkers in one treatment arm, you would expect that if the drug was efficacious, there would be more in that arm than there were in the placebo arm. So it might well be a way of actually salvaging failed drugs or histologically failed drugs by setting the bar much tighter. A great example of this, and we actually looked at this and modeled it, was some of the semaglutide data, where, of course, in the main paper, the histological endpoint for fibrosis was not reached. But when you stack histology, FibroScan, and ELF test and look at only those individuals who have met the predefined threshold for all 3, you actually see a significant difference between placebo and the treatment arms. Now, the N gets very small in terms of the number of individuals who fulfill that, but it implies to me that you are actually moving the needle on fibrosis.
But that's exactly where NAIL NIT can help, Quentin, because it's going to combine that data set with another data set, with another data set, and your specific comment can be addressed across a broad swath of studies. Thanks for pitching.
Well, what are we waiting for? What are we waiting for? Let's get on with this.
That's Jörn's project.
So one last thought. Well, we're advancing. Let's put it that way. One last thought I want to ventilate in this group quickly is, Quentin, you talked about imaging and blood-based biomarkers. Now, we do have quality of life biomarkers that are typically explored in the studies, and we report on them separately. Would this be something we could link in here? I mean, if we report on those trials, we normally compare them with histology, but could we add this in a dimension that's worth and helps us, or any thoughts on that?
So I think that's an interesting area. I think you'd need to consider, Jörn, what you wanted to combine it with. So, for example, if you look at some of the work that Zubair has published, you would say that quality of life goes with fibrosis. If we look at some of the work that you and I published, we'd say, well, also it can go with steatohepatitis. So you'd need to think about what you stack it with, because clearly there's some disparity into what biomarkers you'd put them with. But I certainly wouldn't dismiss it. I think it's a really important facet of the disease state. And maybe if you want to take this to and I don't think we're ready for this yet, but because we don't have the biomarkers to go with it, but the ultra holistic approach, then it feels, functions, and survives. It feels biomarkers and maybe outcomes, but outcomes take a long time. Let's stick with the stuff we can measure in a test tube.
It's an interesting question that's about 3 or 4 steps up the road from what you're talking about right now, but it goes with Jörn's question, whether in fact in different situations, the stacked quality of life is different for, say, fibrosis than it is for steatosis, or is different in one set of patients than another.
Yeah, wouldn't that be interesting to know? And it speaks to the idea of the lived experience of the disease. It speaks to Louise's point about how can we personalize medicine to subsets of patients. What we're talking about here, what we spend most of the time talking about, is how can we simplify and make more robust trial endpoints. But maybe we can apply this and answer much bigger questions that matter even more to the patients we're here to look after.
That's how, Quentin, it's always been my belief that every time you improve the quality of the building block, you improve the quality of what you can build. So that makes sense. Before closing comments, last roundup, any further questions? Louise, that was great. Thank you. Where would you like to be on, each of you, where would you like to be on this issue in 12 months that you think is A, realistic, and B, brings the maximum value? Brave one, go first.
I'll tell you where I'd like to be. If we focus on that stacking and the commonality one, I would have liked to have tried that in a number of studies. I would like to have looked at it in some of the studies, phase 2 studies, where we've had positive results. And I would like to look at it in some of the studies where we've had negative results, and to see what came out, how it shook down. I think that would be the first thing to do. Stephen's point about can we use it to guide us in selecting what the biomarkers we put into our fixed biomarker basket are, I think that would be the next thing. So I'd like us to move in those 2 directions. And where I'd really like to be in 12 months, and this is a dream, is not using histological endpoints in our trials, but moving to non-invasive.
We'd all like to be there, and 12 months sounds a tad aggressive, but good for you.
Just a smidge.
The only thing I'd add to that, and I think that's excellent, I definitely want to be there as well, is could we step back and define a length of improvement in a biomarker that correlates with sustained histopathology improvement? Because that's something we haven't looked at before. As Quentin started off, just because you drop ALT in 4 weeks by 17 units per liter, that doesn't necessarily mean fibrosis got better. And I think if we went back and mined our retrospective data and looked at a drop in a biomarker that happened at 36 weeks versus 24 weeks versus 12 weeks versus not at all, and we looked to see what happened with histology amongst those 4 different cohorts of patients, I can't help but imagine that there is going to be a difference. And then what we have to do is just say, what's the right length of time for a biomarker to have change at a certain threshold? That would be incredibly helpful to the regulatory authorities, to the payer, to the physicians, and to the patients. At the end of the day, if we could define what that length of improvement is that matches a threshold change, that's what I would just add to Quentin's thoughts.
And my look to the stars expands that by saying I'd like to see all these data sets come together, as Stephen has pointed out. NIT did that, or aims to do it, is in the process. Litmus has done so in the past, and by integrating some of the clinical trial data and really looking at this across multiple studies to just broaden the data set and understand, get some themes that we haven't been able to see in a let's say 1,000 patients, but look at them more broadly.
Louise?
I was going to take it in a slightly different way, and in that we can get some commonality between the trials, because at the moment we're all pulling in different directions with different markers, different levels of those in a lot of the areas. So therefore it's really, really difficult for the FDA or anybody to make a decision if we can't actually make a broad decision across and provide the same data set for the same trials each time. I think that strengthens our argument. So to be able to drill down and get a set, as Stephen alluded to, you can fix your prices, you can do everything and get that commonality. I think that drives that data, that drives everything, and I think it just the sooner we get all of that together, the easier it is to prove the outcome and to get the FDA to see the same commonalities between everybody.
Yeah, I think all that's great. This might be a really naive comment, or not entirely accurate, but having now watched years of drugs get shot at the end of phase 2B or not get shot when they should have because there was a way to interpret them into phase 3, it would be great if any of this could improve our ability to determine either how we set up our 2Bs or what came out of 2B that was going to succeed in 3. Because I think, Stephen, when you talk about why money doesn't get invested, one of the reasons it doesn't get invested is because there's a relatively low faith right now on the part of investors that we know what's going to make a phase 3 trial go. And simply the ability to improve the confidence in that prediction. I mean, if you think back, Ella being the obvious one that comes to mind, or actually, in fact, the Intercept also, where phase 2B had challenges, but then we found, or the companies found a way to interpret it into phase 3, and then that didn't make the challenges go away. It just enabled them to see one more set of cards. If we could increase the confidence that people had by how we amalgamate the phase 2B analyses, but what's going to work in phase 3, I know we're not going to get there in a year, you don't have the data, but improving confidence in a phase 3 trial on the way in would be hugely helpful to investors, and as a result, I think the companies. And this feels like a path to get there. All right. I'm going to let everybody go before my voice completely collapses. I promise I will be in better throat next week because this appears to be a fairly mild case, and I'm just not going to talk for the next 6 days except when I need to. Quentin, thank you so much for joining us. Stephen, Jörn, Louise, as always. I don't remember right.
Yeah, hope you have a speedy recovery. If you need me to call you in Paxlovid, let me know. I'm happy to do so.
I thought about doing that last night. I'm going to give it another 24 hours because right now it feels like this is really mild. But if it isn't, that's exactly if your phone rings from me, you'll know that that's what I'm looking for. Particularly if it has a pharmacy name and number attached to it. Thanks, folks. See you all next week, except Quentin. I'll see you next week in London. Thanks. I'll be listening.
Thanks, Quentin, for joining us.
Thank you so much, everyone.
Good seeing you, Quentin.
And now for the Season 3, Episode 26 Business Report.
Hi, as you can hear, this is Louise, not Roger. As he reported on Monday, he's dealing with COVID-19, unfortunately. However, he seems in good spirits, but his voice gave way right after our episode on Monday evening. So I get to add the role of business section newsreader to the other roles I've played on this podcast so far.
If there is a good week for Roger to lose his voice, this is the one.
On Thursday and Friday, Rachel Zayas and I will be attending the 5th Global NASH Congress in London, interviewing some of the speakers and sharing our impressions of the Congress. The episode next week will consist largely of the interview segments and our commentary of that conference.
There were 2 casualties of Roger's COVID-induced laryngitis.
Last week, Roger talked about having cost metrics. By late last week or early this week, you would think he could program his computer without a voice, but I guess that's Roger. Also, the schedule was to complete voiceovers for the first episode of the Special Diabetes podcast last Monday. That did not happen. As a result, we will put the episode on the website as soon as Roger has the voice necessary to finish the post-production. Other than that, all is well, and if Roger's unable to speak enough to host next week's podcast, I'll happily fill in.
Our weekly download totals keep climbing.
For the week that ended Tuesday, we had 1,976 downloads and over 6,000 for the 28-day period. Both of these numbers rank second on our all-time podcast lists. So fantastic work, everybody, and keep downloading. The Vault yielded no secrets this week. In fact, every episode and every conversation in the last 2 months received at least 10 downloads, and virtually no episode more than 2 months old received that many. And with that, I'm off. Roger would thank Magic Mike, Eric, Steve, and Murph the Magnificent for everything they do, particularly this week. And in closing, we all want to thank you, our listeners, whose support, feedback, and kind words keep us energized. So stay safe, surf on, and we look forward to seeing you again next week on the podcast. Bye for now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.